everyone. My name is Vazquez, and I'm currently a senior at UNH Manchester studying personal genomics with Dr. Prescott. This is my final project, and I have decided to really give everybody an introduction to genomics. While studying genomics, I really thought that the base was like the most important part. You really need to have a clear understanding of genetics and basic concepts of genomics in order to really be able to expand your knowledge as the course goes on. So that's one of the main reasons why I decided to focus on this, because in the end I felt like this chapter really helped me succeed in the course and really helped me expand my knowledge in general. So I hope you guys find this interesting, and let's dive right into it. So one of the most important things about genomics, it has it in the name, is the genome. So the genome is something that we really focus on because it's so important to everybody in this world. So what is a genome? It is the entire sum of the genetic components that make up any living organism. So as you can see, this is why it's so important. In other words, it is all the genetic stuff in quotation marks that defines an individual. This is why it is so important, because it makes us who we are, and if we understand the genome, we really understand a lot of things about humans, about other species. That's why it's taken scientists years to finally sequence the genome, and thank God we are getting better and better at it as every single day goes on. So, the genome. The media often refers to the genome as a series of genes unique to an individual, but the genome is much more than that. People, as you know, in the media oversimplify things, which is fine to make people understand, but it is really just so much more than that. There is so many things that you can talk about when you talk about the genome and sequencing the genome and learning more about all the genes that make up a human, like some of you may know, personalized medicine. It's all these things that we're going into in the future, and genes are so important that we really need to understand what these concepts are in order to be able to grasp further innovations within the human society. So only less than 2% of the human genome is known to code for genes. So only 2% of that genome right now, we know it's definitely genes. And the remaining 98% is considered junk DNA, which is absolutely crazy. What? This is absolutely crazy. But it is a work in progress. The human genome is something that is currently being vastly studied, and scientists are learning new information every single day. It's as the PowerPoint goes on, you guys are gonna see that I keep saying, like, yeah, I'm gonna tell you about this, but we are still learning about it. Like, every single thing that considers the genome, we're still learning about who we are as human species, and it's just super interesting to be able to embark in this journey with all these scientists. And this class really helped me open my eyes to this whole different world, because it really is a whole different world that needs to be explored. So now that we know that, let's break down. This is to show you a breakdown of the human genome. So this is what you can see. A lot of complicated things, but it's just so interesting. Like, only a couple of these are genes, not all of them. And This is a 2%, this is the 98%. That's how it works. And so in order to understand genes and other genetic concepts, we have to understand the central dogma. So it's an important concept in the understanding of genomics. It is the process of how DNA is transcribed and translated into polypeptide chains. This really helps understand other concepts like gene regulations, because this is how DNA really replicates and makes genes and proteins that are in our bodies that help with certain functions. And transcription is when a particular segment of DNA, as shown in the first picture, the double helix DNA, is copied through transcription into mRNA, which is the second picture in the box, and it says mRNA right next to it. It's a single helix, so it's only one portion of that DNA. And then translation is when that mRNA is translated into an amino acid sequence or a polypeptide chain, which is made out of, which takes these codons and makes them into proteins that have functions, and this polypeptide chains have functions within our body. So it's really just interesting it's a really good concept to understand, and you really need to understand that. So how does the genome work? Having a genome sequence is inconsequential because the gene is not expressed. Biological effects inside and outside the cell will only occur if a gene is expressed. So the gene expression is very important when it comes to genomics. A gene is expressed through transcription and translation, and the final result will make a single protein. So this is what we just talked about in the previous slide, that these two processes will make a single protein, and it will make that gene express, and that's what we need. So the picture below just gives you another insight of that in a little bit more detail, and how it can make different proteins. And you can see at the bottom they can be completely different, or they can be exactly the same. So let's talk a little bit about gene regulation. The process of controlling which gene in a cell's DNA are expressed to make functional products like proteins. So this is a process of which genes do we need for these proteins. Picking and choosing. That's really what DNA does. And transcription factors are part of this because they are a set of regulatory proteins that allow the protein to attach to specific binding regions of the genome, which are really important in the end to specify what genes we want to be expressed and what we don't. So the biology of gene regulation is still an intensive area of research. Like I told you guys before, this is just going to continue. Like, we're still trying to figure this out. We're still trying to learn. So this is just a little picture that shows the levels of protein. So we have a primary, secondary, tertiary, and quaternary. I don't even know how to say that. But that is just how complex proteins can get and how important they can be in the end. Like hemoglobin, very important. You'll learn that in biochemistry. Okay, the human epigenome. The epigenome is an abundance of chemical compounds that can tell the genome what to do. Epigenomic compounds attach to the DNA and modify its function. These compounds mark the genome but do not change the sequence of the DNA. It's very important you make sure you know that. However, they can change the way cells use the instructions of DNA. So they're not changing the sequence or anything. They're just changing kind of like the interpretation, which is kind of cool. So some of the factors that go into this is genomic imprinting. Despite the fact that an individual has two copies of a gene, mom and dad, Not both will be expressed. They should be, but not. Sometimes they're not. And it only affects a small percent of genes, but it can have consequences to an individual, which are things that we need to really think about when looking into gene expression, gene regulation, genomics in general. There's also environmental factors. A lot of people forget about environmental factors, but the environment can work through epigenetic mechanisms to alter human biology. So evolution, like we need to keep these things in mind as well. So then there's genetic variation. This describes the observed variability in genetic and genomic characteristics between individuals and populations. So let's get it a little bit more complex, and there's a term called polymorphism. This term describes the genetic variability that can be found along the human genome at a particular locus. So what this means is that our genes have a locus, and it's very important. And we can find these, and it can cause genetic variability. Humans have about 1% variability between each other. So if me and you were to sequence our genomes, it would be 99% like similar, but there's always that one nucleotide that just changes it. You may have AT, but I might have AC. Just simple things like that that can really just alter the genome and completely change the proteins that are expressed, the genes that are expressed, just everything. And these polymorphisms can come into existence by mutations that persisted throughout history and spread throughout the population. Here we go with the environmental factors again that we do have to keep in mind. These mutations can be caused by errors in DNA replication, radiation, mutagenic toxins, structural methylations, viral activities, and so many more other things can affect these mutations. Let's continue. Polymorphisms, this always gets me, are often inherited together due to linkage disequilibrium. As mutation arise, they are inherited together with nearby alleles. So just right next to it just shows how things can be inherited together. That's kind of just what that picture represents. So linkage disequilibrium refers to the non-random association of alleles at two or more loci. So remember, mutation is random, this is non-random. This brings up an important concept of genetics called SNPs. These are the most common type of genetic variation among people, and each SNP represents a difference in a single DNA building block. So it represents a single difference in a DNA nucleotide. Just like I was saying before, our building block is the same thing. It's those small letters that you saw in the first picture. That was the A and the T and the C and the G. All those. One change in that just can change the data tremendously. So a lot of the data that you see with personal genomics does include SNPs, so it's a very important concept to understand and be able to grasp. But just remember, single nucleotide polymorphisms. So it's just that one nucleotide can just change everything. So that's just a brief overview of what I've learned, and this is the Human Genome Project, which I talked a little bit about, but it was just how scientists tried to sequence the genome, and they finally did. So this is just a timeline of how they didn't. It took a little bit, but it was successful in the end, and that's one of the reasons why we do know a lot about the genes and why we are currently exploring it more and trying to find for new techniques. Yeah, I can make some more later.